about any pathogens. So as I told you, I've worked for 45 years at the Federal University in Rio. So to give you a bit of perspective, and for the last 24 years, we've been trying to work on advanced medicine therapy products, especially focused on the use of stem cells for treating cardiovascular diseases. And as I can tell you now, we totally failed when we went to clinical trials. So the results, basic science, the preclinical science, and the results were very encouraging. But when we went into patients, it really did not work. And after I retired at the university, I was invited by Cruz to try to establish an advanced therapy medicine product program there. And I've been working for the last 2 years with the vice president of production and innovation in health, Marco Krieger, to try to implement this program. And also working with Carlos Morel, who is the head of the Center for Development in Technologies in Health at CDTS, to try to implement an advanced therapy product program at CDTS. So what I'm going to show you is some of the work we've done in the lab. More recently, it involves modeling cardiac diseases using isogenic human-induced pluripotent stem cells derived cardiomyocytes. So we're going to focus specifically on what we call cardiac channelopathies, and those are variants in genes that code either for pore-forming or regulatory subunits of ion channels in the heart. And this can lead to, if you remember your basic science course, to this ECG. This is the atrial depolarization, ventricular depolarization, and this wave is the ventricular repolarization wave. And this can lead to something which is called the prolonged QT syndrome, where you have a prolongation of the duration of the repolarization process, and this can induce severe arrhythmias in patients. And a lot of times those arrhythmias are fatal. So what we've been doing, so to give you an idea, the prevalence is estimated, it's a rare disease. Prevalence is estimated at 1 to 2,000. There have been more than 1,000 mutations related to this syndrome, and they are distributed throughout 15 genes that can lead to long QT syndrome. And here you have the distribution of the duration of the ECG in patients that have long QT and control patients. And so from that, it was established that the cutoff for men is QT interval that's more than 450 milliseconds. And for women, it's a little bit higher. It's 460. Okay? So most here, I show you the 15 genes that can be altered in this long QT syndrome. And but if we concentrate in those 2 genes that code for potassium channels, KCNQ1 and KCNH2, they account for approximately 65% of all the long QT syndrome. And if you add the next one in the list, which codes for a sodium channel in the heart, then you can go up to 85% of all long QT syndromes. So for those three genes, you can explain the majority of long QT syndrome. So all the other genes, they have described mutations, but they're not that important. And why does mutations in those genes induce to long QT syndrome? It's basically because the excitatory process in the heart is basically dependent on a channel, sodium channel current that leads to depolarization of the action potential, as you can see here, okay, in black. This is the depolarization phase. And the repolarization phase, when the action potential, when the potential goes back to the resting state, is basically dependent on an outward current that's carried by potassium channels. Okay? And so if you interfere with any of the potassium channels here, and you decrease the amount of potassium current flowing out of the cell, this would result in a prolongation of your action potential, and that would lead to a long QT syndrome. Okay, so, well, this is, those are embryonic bodies from induced pluripotent stem cells. And those are the embryonic bodies after we submit them to the cardiac differentiation protocol. As you can see, the cells, it's really easy to know if you've been successful because the cells start to contract after 7 to 10 days at the most. And here you can see, I don't know if this is going to play, but probably not, doesn't matter. You can see the fluorescence for troponin T, and here in the flow cytometry, you can see that we can reach really high expressions of this cardiac specific marker. And in here, you could see just the monolayer contracting bodies. So we started looking at this mutation, an arginine to a cysteine mutation in this cardiac channel, and we in this I think it's a 2019 paper, we determined that this mutation actually it was thought that this was a problem of the gating of the channel, but we actually showed that it was not the gating, it was a trafficking defect of the protein to the membrane. And so this was the PhD thesis of Fernanda, who is now at Texas Heart Institute. And so it's long QT syndrome type 2. Okay, the channel is involved is KCNH2, and here we had the proban and a son of this 42-year-old proban, and they both actually had long QT syndrome. Okay, and this can be triggered by auditory stimuli, so it can be triggered by a phone ring or an alarm clock, and as I mentioned before, it can lead to fatal. And we also included in this study a control IPS from a donor in the lab, and we we tried very hard at that time to correct the mutations in the patients, but we failed using CRISPR-Cas. And so what we succeeded was to insert the same mutation in the control line. And that's what I'm going to show you. So to give you a background, this was described by this Japanese group back in 1998. Here is the mutation. It's a missense mutation in a segment of the protein that seems to be associated or may be associated with the gating of the channel. And what this same Japanese group reported in 1999 is exactly that they thought it was related to the gate, but there could be some other mechanism. So this is how we managed to introduce the mutation in homozygosis. Usually those syndromes in heterozygosis, and in doing the CRISPR-Cas in the control cell, the mutation was introduced in homozygosis. Okay, and so here you have the control, the first patient, second patient, and this is the control line that was where we introduced the same mutation in homozygosis. So this is the karyotype, and here is just showing you that the mutation was a heterozygous mutation in both patients, but in the CRISPR line, it was introduced in homozygosis. So here is just to show you that all the pluripotence markers are well expressed in all of the 4 cell lines. Okay, and here is just to show that they all expressed also differentiated markers for endoderm, mesoderm, and ectoderm, okay, which is a necessary control to show that you actually produce those pluripotent stem cells. And this is the result. And as you can see here, this is the recording of the action potential of the control IPS. This is the first patient. This is the second patient. And this is the control line where we introduce the mutation in homozygosis. And as expected, this was the longest duration action potential. And so here we have different durations of the action potential at 40% of repolarization and at 90% of repolarization, which is usually what's used. As you can see, the mutated, the CRISPR-treated cell line was the one that had the longest action potential duration. And it's basically the same thing, but just subtracting this from 40. And here we just used a specific inhibitor of that channel, which is called E4031. And as you can see, once you do this in the control line, you get a very huge prolongation of the action potential. But if you do that in the lines that have the mutation, you don't see any significant prolongation of the action potential because that current is already increased. And so here is how this protein is arranged in the bilayer, okay? And that's the S4 segment, which I told you can be related either to gating of the channel, but there have been other variants in that region that have influenced the trafficking of the protein to the membrane. So we wanted to know what was actually the defect. So we went on and did voltage clamping experiments in single cardiomyocytes. And to make a long story short, as you can see here, this is the activation curve for that current. And you know, it's basically similar all of the four lines. There is no difference in the activation of the ion channel, which means it's not a voltage triggering effect of the mutation. And once we got this result, we went on to look at immunofluorescence for the channel protein, which is the KV11.1. And here you have the control, IPS, okay? And here you have the labeling. This is labeling for troponin in red, labeling for the channel protein in green. And when you merge and you expand this area, you can see that there is labeling at the surface of the cardiomyocytes. But if you do the same thing for one of the patients, you don't see any labeling at the surface. So when we try to quantify this, that's what we got. This is the control. And there is, you know, all the two patients in the CRISPR mutated line, they all have much less protein expressed at the surface, but they have similar intracellular expression of the protein. So it's really a trafficking effect and not a channel gating effect. And so that's the conclusion, that this mutation from arginine to cysteine is a missense variation in the KCNH2 channel that leads to action potential prolongation in the IPS-derived myocytes. It does not affect channel gating, and it is a trafficking effect that reduces channel density at the surface membrane. Okay. So this wasn't supposed to be shown. It doesn't matter. You don't need. No. I'm just now switching to another. This is not yet published, but to be submitted pretty soon. It's long QT syndrome type 1. And that affects different channel, potassium channel, which is KCNQ1. And in this case, we have the proba, which has a QT interval of 516 milliseconds, and the mother. And the mother has the same mutation, but her QT interval is normal. So now it's a more interesting case. We have the same mutation, and the son has the disease, but the mother doesn't. Okay? So this is an increased QT interval. This is the normal QT interval. So this has been classified as a likely pathogenic variant in CLIN4. And there you have the sequence showing that you know, the mother and the son have the same mutation in this KCNQ1 channel. So this is again control, just to show you the IBS. It's a normal karyotype. They all express the pluripotent genes. Here's the immunofluorescence showing that you know, once you differentiate, you have high expression of all three germ layers: endoderm, ectoderm, and mesoderm. Um, and here we start to look at the different behavior of the action potential in the carrier, the son that has the long QT syndrome, and the mother that carries the mutation but doesn't have the disease. So cardiomyocytes derived from IPS from the two patients, they reproduce the phenotype that you see in the patient. And so this is usually called incomplete penetrance. And the question is why you do have incomplete penetrance. So there may be genetic, environmental, or epigenetic causes. And we went on to see if we could dissect this. And so we used a lot of bioinformatics to search for what are called damaging modifiers, which, if you have the same mutation, those can make the mutation worse. Okay? The phenotype is increased in this case, or for protective modifiers that you can have the same mutation, but they protect you from the phenotype. Okay, and so we looked at those various possibilities, and we thought that this SNP in the sodium channel gene could be the culprit. Okay, and this was all in silico. And so investigating the damaging modifier gene, we looked at the proband. Okay, he had two SNPs in this sodium channel. One was a histidine arginine, and the second one was a serine to tyrosine in those positions. And the carrier had only this SNP, but she missed the histidine to arginine. Okay? And so we started to look if this could, be responsible for the incomplete penetrance in the carrier. And in looking at the literature, this paper published in 2005, they showed that control people, normal people who have this SNP, is 588 arginine, it's easier to find in those in population a longer QT interval than in people who do not have that SNP, even though they have a normal QT interval. They don't have the disease, but in terms of population study, they usually have a longer QT interval than people who do not have that kind of SNP. So what we did here is, again, a comparison of the carrier, of the proband who has the mutation and the phenotype, and of the carrier who has the mutation but no phenotype, the son and the mother. And so the first thing we did is we corrected the mutation in the proband, in the son. And as you can see here, when we do that, we do have a decrease that was significant, although not as great as the mother, but we see a decrease in the duration of the action potential of the proband, the son. Okay, and so this indicated that actually this mutation in KCNQ1 was in some degree responsible for the long QT that we observed in the patient. So what we did next is that we then introduced the SNP in the sodium current, in the sodium channel, in the carrier. So the carrier is the mother. She didn't have a phenotype, as you remember. And now we introduced this, his 558 to arginine mutation by CRISPR-Cas, the mother. So, and as you can see here, clearly, we saw an increase in the duration of the action potential. Okay. And leading us to think that this might explain the divergence between proband and carrier. So this is just a summary of all the findings that we have. This is the action potential duration at 90% repolarization of the carrier. This is the carrier that has been where we introduced the mutation in the, well, not the mutation, the SNP in the sodium channel. This is the proband, the sun. And here's the proband when we corrected the mutation in the potassium channel. So we concluded that it is, this mutation is a likely pathogenic mutation, and that this SNP in the sodium channel is a damaging modifier for the long QT phenotype in the proband. And we like to think that we're trying to do precision medicine, and that this is the sort of type of sophisticated diagnosis that will allow us to really see to do precision medicine in cardiac patients. And finally, this is the group. Adriana is mainly responsible for the most of the data that I showed you, along with Thais Brunswick. Those are the collaborators at the National Cardiology Institute. And this is the whole group that has the work. Thank you.
Thank you, Professor Antonio, and also is quite interesting, and also with some details using the actually the method, yes, as a method, and to see the mechanism, especially the congenital disease, and to see which mutation contribute to the kind of disease. So I think it's also, we're open for the question and answer. If I may, questions, please raise the hand. Actually, I have a question. I think in this issue of CNS paper, I think you noticed that the Deng Honghui in China, a scientist, using the iPS-derived isolate to treat the one disease. So I'm wondering, because isolate is also in our body, and the opportunity to counter or interact with outside, especially for the microbiology, the opportunity is less than other cells. So I think maybe the epigenetic modification of these cells is relatively low. So for the cardiac disease, cardiac cell mouse size, I think it's similar. So I'm wondering whether the iPS-derived mouse size in some ways is also going to use to treat this kind of heart failure, or also something. The disease is like isolate can do.
You mean if we can model heart failure using IPS-derived cardiomyocytes? Yeah, no, actually we can. Didn't show you any data, but we've done some work with cardiac hypertrophy that usually leads to have failure. And we can derive IPS from those patients, and we have a major focus on ion channels and electric currents through the membrane. And those hypertrophic cardiomyocytes also exhibit that kind of duration in electrical activity. But other labs have shown that also contractile function is impaired. And we have also produced IPS cells from a very rare disease called progeria, where people age very rapidly. So by the age, they don't live over 15 or 17. I think the longest lived person was a little bit over 20. And we also study, they usually die either of arrhythmias or myocardial infarction. So we also generated IPS from them and corrected by the patient by CRISPR-Cas, and it's actually totally necessary to have isogenic lines to be able to do this type of work, because if they're not isogenic, it's very hard to compare. And the two slides that I that I did not show you is that even when they are isogenic, you can have a degree of variation that's batch. So what we're doing now is that we are correcting the mutations, we are differentiating the pairs at the same time and for the same duration, and then we're doing the recordings also paired, because what we notice is that if you don't do that, the amount of variation that you get in action potential variation can lead you to really false results. So it has to be isogenic, and more than isogenic, you have to control for batch effects.
Yes, yeah, I agree. I think the most difficult part of the clinical application of the iPS-derived cells is the quantity of also the quantity and the quality of the cells, to compare and to simulate the human cells as most as possible. That's I think maybe in the near future there will be some application for heart disease corrections. And I think if we don't have more questions, let's thank Professor Antonio again. Thank you.